Rosalind Franklin was a British biophysicist whose work was instrumental in the discovery of the structure of DNA. Despite her crucial contribution to this landmark scientific achievement, Franklin did not receive recognition for her work during her lifetime. Born in London in 1920, Franklin was the second of 5 children in a Jewish family. She showed an early aptitude for science, and after completing her education, she went on to pursue a career in research. She obtained a Ph.D. in physical chemistry from Cambridge University in 1945 and then moved to Paris, where she worked at the Laboratoire Central du Services Chimiques de l'État. In 1951, Franklin returned to England to work at King's College London. It was here that she began her work on the structure of DNA, using a technique called X-ray crystallography. This technique involves shining X-rays through a crystal of the substance being studied and measuring how the X-rays are diffracted, which provides information about the structure of the substance. Franklin's X-ray crystallography images of DNA were key to determining the double helix structure of the molecule. In particular, her Photo 51 image, taken in 1952, showed a clear X-shaped pattern that indicated the helical structure of DNA. This image was shown to James Watson and Francis Crick, who used it as a key piece of evidence in developing their model of the structure of DNA. Despite her crucial contribution to the discovery of DNA, Franklin was not included in the Nobel Prize for Physiology or Medicine that was awarded to Watson, Crick, and Maurice Wilkins in 1962. This was because Nobel Prizes cannot be awarded posthumously, and Franklin had passed away from cancer 4 years earlier. However, many scientists and historians have argued that Franklin's work was critical to the discovery of DNA and that she should have been included in the Nobel laureation. Watson and Crick did acknowledge Franklin's contribution to the discovery of the structure of DNA in their initial publication in the journal Nature in 1953. In the paper, they stated that they had been stimulated by a knowledge of the general nature of X-ray diffraction photographs of DNA and cited 2 references to Franklin's work. However, Watson and Crick did not mention Franklin by name in the paper, nor did they acknowledge her contribution to the interpretation of her X-ray crystallography images of DNA. Instead, they relied on a general reference to unpublished work as the source of their knowledge of the general nature of the X-ray diffraction photographs of DNA. After the publication of the paper, Watson and Crick continued to downplay Franklin's contribution to the discovery of DNA. In their subsequent papers and talks, they emphasized their own theoretical and conceptual work while minimizing the importance of the experimental data provided by Franklin and others. In addition to her work on DNA, Franklin also made important contributions to the study of viruses and coal. She was a dedicated scientist who was known for her meticulous attention to detail and her commitment to scientific rigor. This can be interpreted as sexism against women during that period of time. Women scientists in the mid-20th century faced significant barriers to recognition and career advancement due to pervasive gender bias and discrimination in the scientific community. Franklin's experience is a prime example of this. Despite her critical contribution to the discovery of the structure of DNA, her work was often overlooked and undervalued by her male colleagues. She was subjected to condescension, patronizing comments, and even outright hostility from some of her male colleagues, who saw her as an outsider and an interloper in the male-dominated world of science. In addition to the challenges she faced as a woman in science, Franklin was also working in a highly competitive and high-pressure environment, where scientific breakthroughs were often the result of fierce competition and intense rivalry between research groups. In this context, it can be difficult to disentangle the effects of gender bias from other factors that may have contributed to Franklin's exclusion from the Nobel Prize. However, It is clear that gender bias played a significant role in limiting the recognition and career advancement of women scientists in the mid-20th century. Many talented women scientists, like Franklin, were denied the opportunities and recognition they deserved due to systemic sexism and discrimination in the scientific community. It is only in recent years that their contributions have begun to be fully recognized and celebrated. Tragically, Franklin's life was cut short by cancer at the age of 37. Franklin's work on X-ray crystallography was critical in providing important data on the structure of DNA, but it also involved exposure to high levels of radiation, which may have contributed to her illness. In addition, Franklin's cancer was not diagnosed until it was in an advanced stage, which unfortunately was not uncommon at the time. However, her legacy as a pioneering scientist who made important contributions to our understanding of the structure and function of DNA continues to be celebrated today. Despite the fact that her name was not included in the Nobel laureation, Franklin's work on DNA remains a testament to her scientific brilliance and her unwavering commitment to the pursuit of knowledge.